What is a bacterial growth curve? What are the procedures for obtaining a bacterial growth curve? And what are the different stages or the different phases of bacterial growth curve? If you want to know all these things, then watch the video till the end. So welcome to a new video of AVIJIT'S biology, and in this video I am going to discuss about the bacterial growth curve, and I will discuss about the different phases of bacterial growth curve. First of all, we have to know what is bacterial growth. So basically bacterial growth is a complex process that involves numerous anabolic and catabolic reactions which result in cell division, and it is proliferation of bacteria into two daughter cells in a process called binary fission. And actually binary fission is a process where a single bacterial cell produces two daughter cells, and the formula of the production of daughter cells is 2 to the power n. Means from one cell there will be production of two cells, and from the two cells there will be four cells, then from that four cells there will be 8 cells, so on. Now we will see what is bacterial growth curve. So the bacterial growth curve represents the number of live cells in a bacterial population over a period of time. And there are 4 distinct phases of the growth curve. The first one is the lag phase. Second one is the exponential or the log phase. Third one is the stationary phase. And the final one is the death phase. Now we will look into the methods of obtaining bacterial growth curve. So first of all, a population growth curve for any particular species of bacterium may be determined by growing a pure culture of the organism in a liquid medium at a constant temperature. Then samples of the culture are collected at fixed intervals like every 30 minutes, and number of viable organisms in each sample is determined. Then the data are plotted on logarithmic graph paper, and the logarithm of the number of bacteria per milliliter of medium is plotted against time. So if we take the log of number of bacterial cells in the y-axis and the time in the x-axis, then we will find a curve like this, which is actually the bacterial growth curve, where this is the lag phase, this higher peak is the exponential or the log phase, and this one or this straight line is the stationary phase, and this declined area is the death phase. So a complete bacterial growth curve will look like this. Now what is lag phase? Actually the lag phase is the initial phase of the bacterial growth curve, and this initial phase is characterized by cellular activity but not growth. A small group of cells are placed in a nutrient rich medium that allows them to synthesize proteins and other molecules necessary for replication. These cells increase in size, but no cell division occurs in this phase. So there will only be the cellular activities, but there will no growth of the cells, and the duration of this phase is 1 hour to several days. Now next one is the exponential or the log phase. So this period is characterized by cell doubling. The number of new bacteria appearing per unit time is proportional to the present population. If growth is not limited, doubling will continue at a constant rate. So both the number of cells and the rate of population increase doubles with each consecutive time period. For this type of exponential growth, plotting the natural logarithm of cell number against time produces a straight line, and the slope of this line is the specific growth rate of the organism, which is a measure of the number of divisions per cell per unit time. And the duration of this phase is few hours to several days. Now the third phase, which is the stationary phase. The stationary phase is often due to a growth limiting factor such as the depletion of an essential nutrient or the formation of an inhibitory product such as an organic acid. Stationary phase results from a situation in which growth rate and death rate are equal. The number of new cells created is limited by the growth factor, and as a result the rate of cell growth matches the rate of cell death. Means there is number of cell growth equals to the number of cell death. The result is a smooth horizontal linear part of the curve during the stationary phase. Mutations can occur during this stationary phase, and the duration of this phase is few hours to several days. Now the final phase of the curve is the death phase. As nutrients become less available and waste products increase by time, the number of dying cells continues to rise. In the death phase, the number of living cells decreases exponentially and population growth experiences a sharp decline. As dying cells lyse or break open, they spill their contents into the environment, making these nutrients available to other bacteria. This helps spore producing bacteria to survive long enough for spore production, and the duration of this phase is few hours to several days. Now I will discuss about the significance of bacterial growth curve. The study of bacterial growth curve is important when aiming to utilize or inoculate known numbers of the bacterial isolate. For example, to enhance plant growth, increase biodegradation of toxic organics, or the production of antibiotics or other natural products at an industrial scale. Here, the knowledge of bacterial growth kinetics and bacterial numbers in a culture medium is important from both a research and commercial point of view. Growth kinetics is also useful for assessing whether particular strains of bacteria are adapted to metabolize certain substrates such as industrial waste or oil pollution. Bacteria that are genetically engineered to clean up oil spills, for example, can be grown in the presence of complex hydrocarbons to ensure that their growth would not be repressed by the toxic effects of oil. Similarly, the slope and shape of growth curves produced from bacteria grown with mixture of industrial waste products can inform scientists whether the bacteria can metabolize the particular substance and how many potential energy sources for the bacteria can be found in the waste mixture. So this was all about the bacterial growth curve. Hope it will help you. So thank you for watching the video. Please like the video and share the video, and please subscribe my channel for such videos. So thank you. We'll meet you in the next video.